Now we are going into another basic unit in biotechnology. It comes under primarily both tools as well as in processes of biotechnology, and the name of the chapter is termed as polymerase chain reaction. Now once we have taken the DNA, practiced use of restriction endonucleases, then later on we have cleaved the sort of a DNA, we have our gene of interest, we have loaded the gene of interest, put it in cloning vectors, and we have again sent it into the sort of a bacteria. Now once it is present in the bacteria, the gene of interest need to be taken and we require multiple copies of the gene of interest. The gene of interest is a very small microgram of the sort of a DNA. Now we have an innovative technique which is termed as polymerase chain reaction. Now what is the goal of polymerase chain reaction? In 1985, Mullis, who was awarded the Nobel Prize, developed a revolutionary innovative technique which is termed as polymerase chain reaction. So in polymerase chain reaction, what do we actually do? We amplify or make multiple copies of the gene of interest with the help of certain biochemical tools and biophysical tools. An analogy would be is, how do you produce 10,000 copies of a paper in a Xerox machine? So here we have primarily the gene getting amplified. We amplify a single sequence of the gene. Thereby the desired DNA, the single copy of the desired DNA is got by the technique of PCR. Now a single PCR, primarily the polymerase chain reaction, the principle of the PCR is based on 3 steps. We have the first step which is termed as denaturation. Now denaturation of the DNA primarily means that we need to separate the 2 DNA strands. After we separate the 2 DNA strands, we would again be annealing or joining the sort of a 2 DNA strands. The second step is termed as annealing. And the third step is termed as extension, or it is also termed as polymerization. So when you take your polymerase chain reaction, the polymerase chain reaction includes 3 steps. Step number 1 is termed as denaturation, step number 2 is annealing or joining, and step number 3 is termed as extension or polymerization. Now we are into first, what is the meaning of the term denaturation of the DNA? That is, we have your target DNA, and at a very high temperature of 94 to 98 degrees centigrade, at a high temperature we would be separating both the strands of the DNA. So in denaturation there is separation of the 2 strands of the DNA. The 2 strands of the DNA remain separated, and here what happens is a single strand, so you have got 2 strands, the 2 strands get separated and a single strand acts like a sort of a template for the synthesis of another sort of a DNA strand, and the entire process has to occur within 2 minutes. So, 2 minutes, 94 degrees centigrade, we are separating this out of a DNA, and this entire process is termed as denaturation. Next, what do you understand by annealing? Now, annealing means we primarily hybridize the two strands of the DNA or the two single stranded DNAs. We have the single stranded DNA. So, we have the DNA at one stage, it gets separated into two strands, and again here we tend to take another sort of strand and we try to join it. So, joining of two primers, there are two important chemicals which are termed as primers. Now, what exactly are primers? Primers are synthetic oligonucleotide sequences which can hybridize at two ends of the sort of a template DNA. Supposing we have a template DNA, in the lab we can manufacture or we can synthesize two important primers. The primer is a sort of an oligonucleotide. They are synthesized. They are added to the PCR, added to the polymerase chain reaction. They are added at both the ends. So, it is just like you are setting up two trains to go in opposite directions. You have your template DNA, onto the template DNA you are running one train and another train, that is you are adding two primers. So, primers primarily complex, they form a complex at the end of the sort of a template DNA. Now, this sort of a process wherein you add the primers, this occurs at a very low temperature, and primarily the temperature at which it occurs, it depends upon the length and sequence of the primer, and the cooling here, permits annealing of the primers or the joining of the primers. So, the joining of the particular sort of a primers, it occurs at a very low temperature. So, this entire process is termed as annealing, and the time which is permitted is 1 minute. So, in a PCR we understand that within 2 minutes the two strands separate, and within one minute we have the primers running on opposite directions, and the primers primarily they use a certain other sort of a nucleotides. And it helps in joining of the sort of primers to the DNA. Now, the third step is termed as extension or polymerization. Now, in extension or polymerization, we have a very important enzyme called tag polymerase. Thermus aquaticus, it is derived from the microorganism called Thermus aquaticus. So, tag polymerase is a sort of an enzyme which primarily is added over here, that is, it is resistant to very high temperatures. And tag polymerase is an enzyme which has been derived from a sort of a thermophilic bacteria. Which is present in the hot springs in thermal aquifers. So, it synthesizes, the tag polymerases synthesizes DNA region between the primers, and during the synthesis it primarily requires magnesium. And it uses DNTPs. Now, DNTPs are deoxynucleoside triphosphatates. So, here we understand that the primers will use the free nitrogen bases and they will be extending towards each other. They will extend towards each other by using DNPs, free DNPs in the presence of magnesium and also in the presence of tag polymerase. All three are needed over here. So, primers extends towards each other and the DNA segment lying between the two primers, the DNA segment which lies between the two primers is copied, and it occurs at an optimum temperature, it occurs at a very optimum temperature of 22 degrees. And the time for primary polymerization is 2 minutes. So, in 2 minutes we have the entire sequence finishes off. So, in a PCR cycle or in a polymerase chain reaction cycle, we have the repetition of denaturation and polymerization. These are the two important components which keep on getting repeated again and again. Therefore, here primarily in PCR or polymerase chain reaction, we understand, first thing is on summary, we understand it is a revolutionary technique which has been developed somewhere in 1985. Next, 1993, he was awarded the sort of a Nobel Prize for working on the PCR. And here there is repeated amplification and cooling, there is a repeated heating and a cooling cycle. There are separation of the two strands of the DNA, and there is a use of primers. Again, in the case of the PCR, we understand that we use a very important enzyme which is termed as stack polymerase. It involves 3 steps like denaturation, annealing and extension by using different sort of temperatures. Now, what exactly are the applications of PCR? PCR has got lot of applications in forensic science. If ever there is a crime committed at a particular sort of a place and we have the evidence of the DNA either in the nails or in the hair or in the semen or in the blood or in the sweat or in any of the sort of human excreta or even with relation to any sort of a chemicals which are coming out of the sort of a criminal's body, it is very easy to take in that small piece of the DNA and amplify the sort of a DNA. And again, it has got lot of applications in dermatoglyphics. Now, dermatoglyphics is primarily relates to the sort of an origin and also the evolution of different characters which are inherited. So, here we understand there is a sort of an exponential amplification of the sort of a DNA, and by amplification of the PCR we can make 1 billion copies of the DNA. 1 billion copies of the gene of interest. 1 billion copies of the gene of interest can easily be done. And we are using only specific DNA sequences, and the entire operation of the sort of a PCR occurs within, it is completely automated. It is simple, it is robust, it is automated, and the process is under in vitro. It always occurs in vitro, that is in the lab conditions, and it is completely automated. So, we have primarily in polymerase chain reaction, it is a sort of a compact, thermal cycle involving 3 important events, and the technique is primarily very important primarily for DNA fingerprinting.